The early developments at late 19th until 20th century. Initially, pathologists depended on smear preparations for cytological analysis. One of the key challenges was preserving the natural structure of the cells. Next, in the 1920s and 1930s, this method allowed cells from fluids and samples to be treated like tissue biopsies. For your illustration, in 1896, Dr. Barenberg's technique used clot formation to process body fluids for tissue examination. Between the 1950s and 1970s, advancements in staining techniques, including the development of immunohistochemistry (IHC), greatly expanded the diagnostic power of cell blocks. Researchers began using these methods to identify specific biomarkers, especially in cancer diagnostics. From the 1980s to the present, modern techniques like immunocytochemistry and molecular testing have been incorporated into cell block preparations, further improving their diagnostic capabilities. Today, the cell block method is crucial for diagnosing malignancies, infections, and various other diseases, particularly in effusion cytology and fine needle aspiration cytology, (FNAC). The role of cell blocks in diagnostic and molecular pathology is to enhance the cytopathological interpretation. These cell blocks improve the interpretation of cytological specimens by providing additional diagnostic techniques such as immunohistochemistry, (IHC), and special stains which help to differentiate cellular features and identify specific biomarkers. Secondly, cell blocks are also used to observe the architectural patterns that are critical for diagnosing malignancies. For example, gland configurations, samama bodies, and stromal invasion. Even in low cellularity specimens, cell blocks enhance the diagnostic yield by preserving the cellular architecture and nuclear detail, crucial for identifying features like hyperchromasia, mitotic figures, or nuclear grooves. Moreover, cell blocks are also essential in advanced molecular studies, which include next-generation sequencing, NGS, fluorescence in situ hybridization, FISH, and biomarker testing for targeted therapies. Based on an additional article that I referred to, the authors evaluated the use of CBs for next-generation sequencing, NGS, in solid tumors, and found that CBs provided sufficient DNA for NGS analysis, yielding accurate and clinically relevant molecular profiles. Lastly, it also ensures the nucleic acid stability, because the paraffin embedding process will enhance the stability of DNA and RNA, hence, enabling prolonged storage. I also included a clear picture of a samoma body that are stained with H and E stain using cell block.
Now we proceed to the advantages of cell block preparation. Cell block offers a high cellular yield that enable us to retrieve adequate material for DNA and RNA extraction. Secondly, it enhanced the morphological detail of a cell that assist in distinguishing between benign and malignant cells by improving their tissue architecture and cell arrangement. Thirdly, it is cost-effective and efficient due to minimal requirement for repeated methods of collecting samples. It is possible to process a single sample for several diagnostic techniques. Finally, long-term storage of paraffin-embedded cell blocks enable us to do follow-up tests and retrospective research. These are differences between each cell block technique with their advantages and their disadvantages. The first technique is Sheetum, that involves mixing cytological material with plasma thrombin mixture. The technique retains architectural features of cells and tissue, but it is time-consuming. Second technique is agarose gel, that mix cell with warm agarose gel solution that solidifies and form a gel matrix. The technique provides good cells and tissue structural preservation, but the gel formation causes distortion towards cell morphology. Third technique is histogel, that mix cell with histogel that solidifies upon cooling. The technique is easy to use and handle, but it may cause distortion to the cell morphology. The fourth technique is gelatin foam, that involves mixing cytological material with gelatin solution. The technique provides good cells and tissue structure preservation, but the foam formation may cause distortion on morphology of the cell. The fifth technique is colloidin cell bag, that involves mixing cells with collodion solution that forms thin film when spread on glass slide. This technique retains cellular and tissue architecture, but may cause distortion on cell morphology during peeling process. Finally, alginate technique Cells are mixed with alginate solution that forms gel when exposed to calcium ions. The technique are compatible with IHC staining, but the gel formation may cause morphology of the cell to be distorted. Here are top 3 recommended cell block techniques to be used. The first one is histogel. Secondly, agarose gel technique. And the final technique is Schiedam technique.
Cell block preparation is a critical process in diagnostic pathology, providing the groundwork for advanced testing like cancer marker detection and molecular diagnostics. But the process is not without its challenges. Challenge number 1: low cellularity in specimens. Some samples don't have enough cells to make a good cell block. This makes it harder to analyze and can limit advanced testing like cancer marker detection. Challenge number 2: technical variability. Different labs and technicians may use different techniques, which can lead to uneven results in cell block quality. Things like centrifugation speed or fixation time may vary and affect the outcome. Next, fixation and processing issues arise when samples are delayed or exposed to inappropriate fixatives. This can lead to cell degeneration and reduce the quality of diagnostic and immunohistochemical results. Artifacts in cell block preparation, such as folding, fragmentation, or cellular morphology loss, create further challenges. These artifacts can interfere with both morphological assessments and molecular testing. Another issue: loss of specimen during processing. Techniques like needle rinsing may lead to significant loss of diagnostic material, especially for low cellularity samples. Challenge number 6: lack of standardization. The absence of standardized protocols across laboratories for cell block preparation leads to variability in quality and reproducibility of cell blocks. Inconsistent methods make it difficult to compare results across labs. Time and cost considerations also limit advancements. Some techniques require expensive materials or take significant time, making them impractical for many laboratories. Finally, molecular diagnostics can be impacted. Formalin fixation and paraffin embedding may degrade DNA and RNA, while non-tumor components like stroma in cell blocks can interfere with molecular assays, making them less reliable compared to core biopsies.
Recent advancements in cell block preparation, focusing on 3 key innovations. The first one is next-gen cell blocking device, second one is the subtractive coordinate immunoreactivity pattern (SCIP) approach, and the last one is fully automated system. Next-gen cell blocking kits is based on Schiedam method. These proprietary kits utilize preformed media discs with wells and an inbuilt AB marker to control the depth of cutting and help SCIP approach. They accommodate various specimen types, including hypocellular specimens. Here's the example of how Nano next-gen cell blocking works. Next is subtractive coordinate immunoreactivity pattern. In cell block preparation, this approach is a method used to enhance diagnostic accuracy in histopathology and cytology. It involves subtractive analysis of immunoreactivity patterns by using multiple immunohistochemical (IHC) markers in a systematic and coordinated manner to identify specific cell types or disease states. This approach works by applying a series of IHC stains to cell block samples, observing how specific markers are expressed, and interpreting their interactions to distinguish between similar-looking cells or pathological conditions. Subtractive analysis eliminates non-relevant signals, focusing only on diagnostically significant patterns. The benefits of SCIP include improved precision in diagnosing complex cases, better differentiation of cell types or tumors, and enhanced reproducibility of results. It speeds up and simplifies the immunocytochemical evaluation of effusion fluids in all cases, making it an essential tool in modern pathology. Serial sections of cell block allow evaluation of immunoreactivity by different immunomarkers in same cells. The latest advancement is Celliant Automated System. It is an automated cell block technology that uses vacuum-assisted filtration to capture and concentrate available cells for maximum cellularity, even from small or scanty samples. The Celliant CB system is a suitable technique to combine with gynecological cytology as they produce high-quality cell blocks, as you could see in the comparison picture. Based on study by Xing et al., Celian CB system is useful in the differential diagnosis of atypical glandular cells in Papa Nicolau tests.
Now let's move to the future directions of cell block. To improve this technique, we need to focus on three keys, which is standardization and automation, better preservation and fixation, and cost and efficiency. First, standardization is crucial because different labs use different methods that leading to inconsistent results. Developing standardized protocol will improve the accuracy. Other than that, automation can also help, but current systems are costly. The goal is to create affordable automated processing system to make workflow easier and more efficient. Second is fixative. Fixative are essential for preserving cell samples, but not all of them work well with every technique. Future research should focus on finding better fixative and embedding material that improve cell structure and analysis quality. Last but not least, cost is a major barrier to widespread use. We need to find cheaper fixative and embedding material. Furthermore, faster fixation technique will also help reduce waiting time and speed up analysis. In short, improving these trees will make cell block technology more effective, accessible, and reliable.